Good morning, everybody, or afternoon, depending on where you're calling us from. Welcome to today's webinar. Today, we will be sharing with you details of our 9 recently funded grant awards awarded by FPWR, which will be advancing our mission to find treatments for PWS. This is Susan Hedstrom, Executive Director for FPWR, and joining us on today's call is John Walter, CEO, and Theresa Strong, Director of Research Programs. Before we get started, just a little housekeeping. We really want to hear from you on today's call, and we want to answer all of your questions. You can submit a question at any time during the webinar using the GoToMeeting control panel. Simply type your question into the question field, and it'll be added to our queue. We will reserve time throughout the webinar to answer your questions. And at the end of the webinar, if you would like to ask a question directly of Theresa, you can raise your hand and we will take you off mute so that you can have a direct interaction with her. So at this point, I'm going to hand off to John Walter, our CEO, so that he can have a brief welcome.
Thank you, Susan, and welcome everyone. As Susan mentioned today, Theresa Strong, FPWR's Director of Research Program, will share the recent round of research projects that we've funded with the generous support of many of you on the phone and countless others from our community. This critical research is not possible without your support, so I'd like to thank you. Before we begin, I wanted to provide a brief update regarding the impacts FPWR is experiencing as a result of the COVID virus pandemic. For those of you on the call and the entire PWS community, is our sincere hope that you and your families have remained healthy and are navigating the economic fallout that has ensued. Managing PWS can be challenging in normal times and can be exacerbated to new levels during times of stress, such as what we are experiencing with the COVID-19 pandemic. To provide you with needed support to help you manage your day-to-day lives with everyone at home, FPWR has hosted several webinars discussing behavior and mental health. These resources are available on our website, fpwr.org/blog. The organization is not immune to the economic fallout resulting from the closing of our economy either. We're currently working with our board of directors, establishing contingency plans for the remainder of the year. Our goal is to maintain a sound financial footing to ensure the perpetuation of the foundation and to continue to support the research that we all know is so important. In a considerable gesture of generosity during this challenging time, an anonymous donor has offered to match up to $50,000 now until the end of the month. At the same time, I fully realize that many of you may not be in a financial position to support FPWR, but if you can, we need your support now more than ever. Thank you all very much. And Theresa, I'll turn the presentation over to you.
All right, thank you, John. So I'm really excited on behalf of the entire FPWR research team to share with you today some of the progress that's been made in our grant program and also some of the new grants that have been funded. So I'll talk a little bit about the ongoing PWS research activities and also briefly on the impact of the COVID on that research activity. Then we'll talk about returns on investments and new publications that have come out with the support of the grant program projects, and then talk about the new grants projects that are funded in 2020. So these are grants that or proposals that were submitted last fall and were reviewed over the holidays and are now coming online. And I think we'll have time for questions and discussions at the end, which I would definitely welcome. So first, just a word about the COVID impact on the research enterprise. So like everything else, a lot of the vast majority of labs have had to slow or in some cases stop research. So the majority of university research labs are allowing the maintenance of, for example, cell lines and animal models that really take a long time to derive. So those are being maintained, but many universities are not allowing new studies to begin until the pandemic settles down or is over. And so there has been some slowing down of work in the laboratories themselves. On the other hand, I think it's been a positive in some ways, in that it has allowed researchers to step back and do a lot of data analysis. Researchers love to do lab work, and sometimes it's, there's a silver lining to this, in that it allows them to step back, do data analysis, write papers, get their grant proposals together, and spend a little bit of time in strategic planning and also writing things up. So that's a good thing. And however, we are hoping that it doesn't drag out too long. And we're already starting to see some signs. I'm getting information from some of our colleagues in Europe that they are going back into the labs and starting up research programs again. So hopefully that will be the case. We haven't heard of anybody so far having any difficulty themselves with COVID for themselves or their families. So we're really grateful for that, and hopefully things can come online pretty rapidly. There's also, obviously been an impact on the clinical trials that are ongoing in PWS in the US. Many of them have been switched to virtual visits or paused or have been delayed in starting. Susan is staying in touch with the sponsors about this, and if you are checking our webpage or if you have signed up for our clinical trial alert, you'll be informed of all of those changes. And when these trials come back online, we'll let you know as soon as we hear from the sponsors themselves. So please keep an eye out for that. I'll also mention that maybe this is a time, since you're home a lot, to update your global registry record. As you may know, the global registry is aimed at understanding the full spectrum of PWS characteristics, and the more complete records that we have in there, the better we can understand the challenges that our adults and children with PWS face. So if you have an opportunity to update your records, we would really appreciate you doing that. We're doing many studies, or have done many studies through the registry, identifying areas of needed research, looking at the patient perspective in the drug approval process. It is a good way, I find, of keeping track of your child's medical history, since there's a lot to keep track of in life, and it gives you a place to store information about medications and medical history. We are developing a survey about COVID, including, how people are coping, what has been helpful for individuals with PWS as they cope with the changes associated with this pandemic. We are, as we speak, that is being established in the registry. So please look for that probably by the end of the month or so. And we hope that you'll have an opportunity to go in and fill that out, so we can just gain an understanding of how our kids are doing, what are their needs, and what's working for them. Okay, so let's turn to the grant program. Even during this COVID epidemic, our research team has been really focused on the one thing that we always focus on, and that is how do we accelerate treatment for PWS and get that to our loved ones. So we have many programs across the spectrum that goes from the research and discovery phase all the way through drug development, and then optimization of clinical care. And some of the programs that support that are shown on the bottom here. But the grant program is a program that supports research across this entire spectrum. So we're really looking for innovative work, exceptional opportunities that can impact any point along this pathway. And you'll see that in the proposals that we talk about today, and also some of the outcomes that we're seeing. So since its inception, we've invested more than $14 million into PWS research, supporting more than 180 research projects in 13 countries, and more than 170 publications have come out of that. And, that's important because that's how scientists and medical professionals share information and build knowledge and move us forward in that pathway of improving the lives of those with PWS. Those publications have really spanned everything from, basic biology of PWS gene function to clinical trials and optimization of care. And while, it's true, $14 million is a lot of money for us as a patient community to invest in this process. But I think it's important to know that those investments beget, more investments from people outside of our community. So The FPWR supported investigators have received now more than $25 million in additional funds from large governmental agencies like the NIH, from venture capitalists, for example, for some of the drug development studies. So our job is really to try to leverage our funds as efficiently as possible so that we can encourage larger projects that can then pull additional funding in from other sources. In addition, we've generated new resources that can be used by the wider PWS research community, as well as new drug targets and candidate drugs and insights into clinical care. So this slide just shows a few of the outcomes that have publications that have come from our grant program in recent months. And I'll just mention a couple of those in a little bit more detail. So first is this paper that comes from Harold Van Bos, who is, many of you see, he's an orthopedic surgeon who takes care of many individuals with PWS. And this is a paper that came out of a project that was initiated actually by a PWS mom, Jessica Cox, and also Marie Gantz, who is a PWS mom and doubles as a biostatistician, also contributed to this project. And it really came from a discussion about hip dysplasia in babies with PWS. So we know that hip dysplasia is really common in infants with PWS, much more common than in the general population. But the long-term consequences really aren't known. In the general population, hip dysplasia in infancy can be associated later on with having more hip and knee replacement surgery. But that didn't seem to be the case, or potentially wasn't the case. But as Jessica was talking with Dr. Van Bos about, about this and about this clinical decision making, they decided to do a project in which they examined this huge database that has millions of records and look at the hospitalizations for hip and knee replacement in adults and compare the typical population to PWS. And what they found in this study is that the incidence of later hip and knee replacement in PWS is actually lower than it is in the general population. So this suggests that watchful waiting or observation by an experienced PWS orthopedic may be the best way to go, rather than aggressive treatment, which can include a halter to that restricts movement of babies with PWS. So this is an example of a PWS parents influencing and working with the clinicians to answer pretty important but straightforward questions about how to care for individuals with PWS. We also saw a number of papers. We have, in the past few years, published or supported several projects about early development of individuals with PWS and kind of understanding that development, social development, and working towards optimizing that. And I'll just summarize that in this slide. It doesn't do each of these papers justice, but we do have much of this information on our website if you're interested in more detail. But just in a big picture, some of the conclusions from these papers are that, the food and non-food related behavior problems that we see in PWS, they differ across settings, as you might imagine. And there tend to be more food-related behavior problems at home compared to school. And this may be because it's harder to keep a schedule at home. School is a much more structured environment. There are also a couple of papers were showing that the parent report, it may not match that of teachers and clinician reporting, and so that it's important to gather information across different settings to really understand how kids with PWS are developing. Some of the work from Dr. Taubert and Dr. Pascal Baron in France looked at how individuals with PWS are processing voices and non-voice sounds, and how they're processing emotions and facial recognition. And they really found some differences in PWS compared to other populations. It takes longer for individuals with PWS to process sounds, and they need to accumulate more information before they'll make a decision. They're particularly challenged in reading facial expressions, so it takes them longer to recognize emotions, and they're less accurate about that. And across studies now, it seems as if those with PWS by UPD, uniparental disomy, that genetic subtype, have a harder time, both with processing the sounds, and they're also less likely to gaze into the eyes, and so they have a harder time reading emotions. And this can make their social development more difficult. So specific deficits in that social cognition or that understanding of social situations can be seen very early on in development in preschool age children, age 3 to 5. It differs by genetic subtype, with the UPD tending to have a more difficult time. And all of these studies kind of in the aggregate suggest that tailored interventions beginning early in child development are really needed and may be beneficial. So I'll talk more at the end about what we're doing. It's great that this information is getting into the medical literature. Our question now and our efforts now are really working with these investigators to develop more practical things that you as a parent can, work on at home or bring to a teacher or bring to a therapist to address these issues that we know are challenges now in our kids. I'll mention 2 more studies, one of which is eye tracking in PWS. Many of you probably participated in this study with the group Sasha Key at Vanderbilt University, and they've published their first paper looking at the ability of eye tracking to measure interest and hyperphagia in PWS. So that will it's the start of a longer process towards improving the outcomes that we have in clinical trials to measure hyperphagia, but it's a good first step. And then another publication that came out recently is from Tony Holland's group in the UK. They were looking at vagus nerve stimulation as a means to reduce problem behavior in PWS, and shown across the bottom here is the incidence of temper outbursts in several individuals with PWS. And what they showed is that with this vagus nerve stimulation, in most of the individuals, the number of temper outbursts really was reduced over time. And for those of us who have an older individual with PWS who is prone to temper outbursts, you know that this can be a really challenging aspect of the disorder. It is stressful for everybody involved, and it can be limiting for those with PWS. It may limit their living situation or their ability to hold a job, for example. So this is a really interesting and potentially promising intervention. And we just yesterday, in fact, had a really nice discussion with reviewers at the FDA, at the Food and Drug Administration, looking at what are the best next steps for really demonstrating in a larger clinical trial that this might be an effective treatment for temper outbursts in PWS, and how do we best approach that. So, so look for some more information about that developing effort in the coming months. Okay, great. I will run through the new grants that we have funded now. I mentioned, Susan mentioned earlier that there are 9 of them, and the list of them is here, but I'll break them up into groups. So the first several grants are really focused on understanding the basic biology of PWS and the genetics of PWS. Our grant program is where we fund this kind of research, and we fund this kind of research because ultimately understanding the pathways and the genetics that underlie PWS may help us be more efficient in identifying new treatments for PWS. So this is the obligatory chromosome 15 slide that shows all the genes in the PWS region. And it's just a reminder that this is a very complex region that has these funky RNAs called non-coding RNAs that are really unusual in the genome. So this is a really challenging area of the genome, and we just happen to end up right in the middle of it. So the first project is really trying to look at that region with a new eye, and this is a researcher from the UK, Dr. Grzechnik, and his lab is interested in understanding how deletion or inactivation of this region impacts this particular class of RNAs called the non-coding RNAs in the region. So their goal is to apply some more advanced sequencing technology to look at these complex RNAs that are generated in that region, and then see what happens when pieces of the DNA are deleted, how those non-coding RNAs change and how they influence other RNAs. So this study applies a new approach to RNA analysis for the aficionados in the audience, where they're using nanopore and Illumina sequencing. And the goal is to understand how the non-coding RNAs function in the PWS region to identify future therapeutic approaches. We're also interested in the study because it brings a new expertise in RNA biology into the PWS field. Dr. Grzechnik is an expert in these non-coding RNAs, but really hasn't done much work in the PWS region. And the long-term goal is to uncover the molecular mechanisms of PWS, thereby identifying pathways that might be good targets for drug development. A second grant in this category is from Dr. David Godler at Murdoch Children's Research Institute in Australia. And this project will look at, again, looking at RNA signatures in blood and brain, and looking at whether those RNA signatures might be predictive of behavioral challenges in PWS. So the Godler lab is interested in identifying early predictors of autism and serious mental illness in PWS. We all know that all of our kids are at higher risk for both of those. So how can we identify those individuals who are at the highest risk and hopefully intervene early? And what will that understanding that help us in the way of developing new therapeutics? So the preliminary studies from Dr. Godler's group suggest that there may be inflammatory pathways that are linked to this process, and that a particular gene in the region, UBE3A, may be important in helping to determine who is going to have the most difficulty. So this study will examine how UBE3A activity and those related inflammatory pathways are associated with behavioral issues in children and adolescents with PWS. And it will be, again, comparing the deletion subtype to the non-deletion uniparental disomy and imprinting centered mutation individuals with PWS. So we're excited about this study because it is also using state-of-the-art technology, potentially to help identify early predictors at a time where we might be able to intervene and prevent mental illness or more severe autism characteristics. And we also, it may suggest new therapeutic targets. So if the hypothesis holds and it is linked to this inflammatory pathway, that will send us down new therapeutic pathways looking at anti-inflammatory medications, potentially to try to head off some of these issues. So we're a long way from that, but I think it will be a good step to see if that is the right direction for us to be going. The next project in this grouping is the role of the placenta in PWS, mapping expression of the PWS genes. And this project is from Anthony Ild at Cardiff University. And so we know that alterations in the placental function during the development of a baby can have profound consequences on the brain development of that individual. And also that signaling back and forth between the placenta and the maternal and the mom can have consequences on the maternal brain as well. So this project is really looking at that basic biology question of, how are the PWS genes in the placenta influencing this process of development? So as a first step towards that, this research team is going to generate a detailed map of how PWS genes are expressed in the placenta, which really has been an understudied area of investigation. And they'll be using mouse models of PWS. So we're excited about this study because it is a brand new area of investigation to understand the origins of the developmental challenges in PWS, studying the genes in the placenta. It's using some novel techniques that were developed in the Isles lab. And Dr. Isles is an experienced PWS researcher. He's done a lot of work in PWS and really understands these mouse models well. And we also have an opportunity here to leverage some limited funds with some support of students in Dr. Isles' lab. So FPWR is going to be contributing to some of the supplies, but the labor, the intellectual power will be provided by some students that are already in Dr. Isles' lab that have other support. So it's a way to train them in PWS and gain some young, smart investigators in the field. So the long-term goal is to identify whether the neurological symptoms of PWS are actually programmed early in development through deficits in the placental function. And this could lead down the line potentially to novel interventions and treatments and understanding of the developmental profile of PWS. All right, the last study in this category is a second year of support for a study from Dr. Larry Reiter looking at epigenetic-driven circadian rhythm defects in neurons from individuals with PWS. So the Reiter lab you may be familiar with, maybe you've sent your children's baby teeth to Dr. Reiter. Their group is using those baby teeth to generate stem cells that can be studied in a dish and look at changes in PWS biology compared to other stem cells. And this particular project is looking at those stem cells and examining the circadian rhythm. So you can actually, that day-night rhythm is actually, can be seen in cells in a dish. And some early studies by Dr. Reiter's group in collaboration with some other PWS investigators, including Dr. LaSalle, have shown that there's a disruption, even at the cellular level, of this sleep-wake cycle, and it can be studied in the lab. So we're excited about this study because it's a great collaborative team. It brings together Dr. Reiter, as I said, with Dr. LaSalle and others who are experts in epigenetics of PWS. It's using a unique resource, these dental pulp stem cells, and it's a new approach to understanding the biology. And if they are successful at really being able to measure the circadian rhythm defect in cells in a dish, it sets them up where they can develop a small molecule or drug screens to see if there are drugs that can reset that rhythm to a more normal rhythm. Okay, we have two projects that are really focused on the neurobiology of PWS, and those are shown here. Both of these are second-year support, so they're projects that we have funded before and are making good progress, and so we are supporting them for a second year. The first is a collaboration between Sebastian Boré and Françoise Muscatelli, looking at the critical periods for oxytocin to have important impacts on behavior in mouse models of PWS. So this collaboration was actually generated based on advocate reviewers who saw that both of these investigators had complementary expertise and were doing complementary projects and suggested that maybe they try to get together, and they have. And they're looking at how oxytocin treatment, where in development it can have its biggest impact, and how we might maximize the beneficial effects of oxytocin. So the goal in year 2 of this project is to use a mouse model of PWS to optimize the effects of oxytocin at different stages and to examine the neural mechanisms by which oxytocin exerts its beneficial effects in PWS mouse models. So we're excited because this continues to foster this important collaboration, which has made good progress over the first year. It's going to answer important questions about when in life oxytocin would be most beneficial in PWS. And it will also help us understand how oxytocin influences food intake, cognitive abilities, and social interactions. So the long-term goal is to optimize oxytocin as a therapeutic strategy in PWS. And the second project in the neurobiology category is from Marcelo Dietrich at Yale University. And their lab has really been leading an effort to understand a particular class of neurons, the AGRP neurons, which are otherwise known as hunger neurons in the hypothalamus. And they have evidence that these neurons are overactive in PWS, and they're looking at them specifically in PWS mouse models. So in the second year of funding, they're going to continue to examine the function of these neurons throughout development in PWS mouse models to understand how the circuits change during development. So we're excited about this project because it's really an excellent young investigator. He's made significant progress in the first year. He is a leader in his field, and he's applying that knowledge to PWS. They are using some innovative technology and novel approaches to do so. And, you know, it's really important to understand what is driving that hunger and that change in hunger over time and development in PWS. And the long-term goal of this project is to define the altered brain circuits in PWS and use that information to explore new avenues of therapy. Okay, our last three projects that I'll mention or that we have funded in this round are shown here, and they all deal with different kinds of genetic therapy for PWS. So the first one is really a different one, a different kind of approach than we've seen before. And this comes from Dr. Cow at Ohio State University. And she's developed this really interesting gene therapy approach to treat obesity, in which it is delivery of a engineered gene BDNF, which is important in neuronal growth and also important in metabolism. So she's developed this gene therapy that targets the brain center for energy regulation, and she is delivering a single dose of BDNF to animals, and has shown that this is a really effective way to impact metabolism in some other models of obesity, non-PWS. So in this project, she's going to apply that technology to PWS mouse models and see if it has a beneficial impact. So we're excited about this study because it's the first study of this novel gene therapy approach in the PWS mouse model by an experienced group that has developed this novel approach. And obviously, this is a critical problem in PWS, the obesity and the metabolic defects and that decreased energy expenditure that we see in PWS. And, you know, in addition to, determining whether this is a productive avenue for therapeutic approaches, one of the benefits of this study is it will really potentially give us insights into the role of BDNF in PWS. So there are some older studies showing that there's a deficiency of BDNF in PWS and, suggesting that it might be a good therapeutic target. And this study will, let us know whether it is a good therapeutic target or not. So the long-term goal is to improve the metabolic regulation in PWS with a novel gene therapy approach. And then the final two grants are really looking at epigenome editing. So these, again, are two different approaches to the same issue of can we reactivate the maternal or the silent genes on the PWS, in the PWS region of chromosome 15. So as a reminder, all individuals, whether they have PWS by deletion or by UPD or by imprinting center, they all have the genes on the maternal chromosome sitting there but silenced. And so FPWR is supporting several different approaches to try to understand how we can best activate those genes and do so efficiently and specifically. So this study from Dr. Mussolino in Germany is going to be looking at one of those approaches, and that is using what they are terming designer epigenome modifiers, so activators, so ADEMs, that are specifically targeting the PWS region. So some approaches that are being undertaken for gene activation may activate lots of genes, even genes outside of the PWS region. An advantage of this approach is that it is meant to precisely activate the PWS region. So Dr. Mussolino and his team will develop these ADEMs to target the specific regions on chromosome 15 and test them in PWS cells in a dish in the lab. So we're excited about this study because it is, a different innovative approach to gene activation studies, looking at the chromatin right in that PWS region, and it may allow for a more specific activation of the PWS region. And in the long term, we're supporting several approaches towards gene activation, and it will be a process of seeing what makes the most sense for moving those forward into the clinic. So this is, one really interesting and exciting way to do that. And the final project that we're supporting in this round of funding is from actually Dr. Iglesias at Duke University, engineering epigenome editing tools for sustained reactivation of maternal PWS genes. So this group, and she is centered in a larger group that includes Dr. Charlie Gershbach, who has really led the field of CRISPR editing and CRISPR gene activation. It's one of the world's leading experts. And this group has been looking at the potential of CRISPR-based approaches for gene activation in PWS. And her group has shown that this epigenetic editing can reactivate maternal genes in the PWS region. And now they're addressing kind of a longer, a bigger picture question. Once you turn those genes on, they have to stay on, right? It probably is not going to be enough to turn the genes on, at one stage. The question they're asking here is, how do we make sure that turning on of the genes is maintained over the long term? Obviously, any gene activation approach to PWS, if it's going to be effective, is going to have to stay for long term. So that's the big picture question that they are addressing here. So we're excited about this study because it's investigating the development of a potential gene therapy for PWS and addressing some of the kind of the thornier issues of a gene activation approach to PWS. This is a smart young investigator in an outstanding academic environment, and so she has all the resources and expertise that she needs to make this project successful. And it addresses a significant therapeutic question that is not only important from a therapeutic perspective, how do we keep these genes turned on, but will also inform our understanding of the basic genetics in the PWS region, the chromatin structure and the function of the DNA modifications of the region. And the long-term goal is to develop a stable genetic therapy that has a lifetime of benefit for individuals with PWS. And you can see in a picture from their lab website, they want to turn the genes on and keep the genes on. So that is a bit of a whirlwind of the projects that we are funding in this round. This slide tells some of what to expect in the remainder of 2020. We hope to see very soon, and we're starting to see signs of a rebound in the lab research activity post-COVID-19. So we're excited about that. As I mentioned early on, I think one of our main goals for the FPWR research team this year is the dissemination of research results. So it's great that all of these things are getting out into the medical literature, but how do we translate that into resources that families can use to help improve the lives of their loved ones with PWS? So materials for families, therapists, for educators on development, optimizing development, and social cognition in individuals with PWS, for example. We'll also continue to see data from the global registry. Thank you to everyone who has contributed to the global PWS registry. Because of you, we can go in and see what 500 individuals with PWS, what at a time, you know, in a certain age group, what their behavior is or what medical challenges they're having. So it's really been a tremendous resource for us to understand PWS. We're sharing that data with many of the clinicians. We're also sharing information that is helpful for drug developers to understand our population and the challenges that need to be addressed. So that has been a fantastic resource and really is available because of your efforts. We'll also see new insights into PWS biology and molecular mechanisms. We talk to our researchers at least once in the middle of their research projects. So we're talking to researchers who are currently funded now. And I think there's going to be a lot of advances in our understanding of the basic biology of PWS over the next 6 to 12 months. So we'll look forward to sharing that with you. And as well, we are starting to get some reads on the clinical trials that have been ongoing in our population. Unfortunately, many of you are aware the Melindo-Zephyr study did not show a significant improvement in hunger, and that was disappointing for everybody. But we do appreciate that. You know, the trial was very well done, and you know, we did get an answer. It was not the answer we were hoping for, but it was a good trial and a you know, a strong answer. So we are hopeful for the other trials that are ongoing that we'll have more positive results coming out of those, and we'll start to look for that. I think the Seleno readout is expected around June, so we're excited about that. And there are other, like the Levo trial is ongoing, and we'll have readouts as soon as they can. Don't have a date yet on that. And also there'll be the start of some new clinical trials. So the Harmony trial will be starting on pitolisant and PWS, so we're excited about that as well. And we will be continuing to interact with the FDA, helping them understand the unmet needs in our PWS community, laying the groundwork by working with all of our collaborators in the PWS Clinical Trials Consortium to advance drugs, to help support clinical trial activity. And eventually, we hope get approval of those drugs and look at issues of payment and reimbursement and insurance coverage. Those are the next things we will be looking towards. So that's all I have. Here are some links that you can stay on top of the research findings that are coming out. You always feel free to reach out to myself or Jessica specifically about the grant program. You know, all of this would not be possible were it not for all of you who are, in many cases, doing the fundraising and doing, you know, all of the challenging work so that we can help move this ball forward. So we really appreciate your efforts. And, you know, if there are concerns that you have or particular areas of interest that you'd like to discuss, of course, we definitely welcome those. So Susan, I'll turn it back over to you.
Fantastic. So we do have questions that have been sitting in the queue, and we'll continue to accept those questions coming in. Again, if you would like to ask a question directly to Theresa, you can raise your hand. I'll go back and forth between the two so that people can be taken off mute and ask Theresa questions. However, I do have a number of questions that have been sitting here for a while. So let's go ahead and get started with those first. The first question, actually I can answer this one, is in regards to the pitolisant study and when will it begin recruiting. So Harmony was intending to begin the study this May. However, given the current environment, they cannot begin recruiting at this time, but they do have plans to open the study as soon as the environment allows. There is a letter from Harmony listed on FPWR's blog if you would like to get more details or a message from Harmony themselves, but the study will go on. They just need to wait until it's safe for them to conduct it. They do want to keep all of our patients in good health. The next question, Theresa, was in regards to Dr. Godler's study. That was the second study that you mentioned. Will he be recruiting patients for that study?
He will, but in Australia. So so they have a pretty good network there, I think. So I believe he's going to be recruiting in Australia.
All right. The next question was in regards to, this is Project 4, the circadian rhythm study. Has circadian rhythm been successfully treated in other syndromes or diseases? Have we been able to normalize that circadian rhythm for others?
That is a great question, and I don't know the answer to that, but definitely we should look at that, shouldn't we? Yeah, there are known disorders of circadian rhythm, and I think it's treated pretty like symptomatically with drugs that are used for daytime sleepiness and that kind of thing. But that's a great question, and I'm going to follow up on it. So thank you, but I don't have an answer right now. But it would be important to look at.
Sure. All right, we've got a number of questions in here that are related to the genetic therapy studies that you mentioned. These were the last 3 studies that you presented on. This is a great question. I hear it a lot, is for the maternal reactivation strategies, if you have a child with UPD, will both of these chromosomes be turned on, and is that a good thing?
Again, a great question, and is actually, I don't know if you've seen it, but we have a list of sort of feasibility questions about gene activation, and that's one of our feasibility questions, right? So you could imagine that turning it on, there is going to be a difference between those with deletion and those with UPD. Essentially, you can turn on a double dose potentially in those with UPD, and that may be a good. That may be good because you have kind of 2 possibilities to turn it on, and gene therapy and gene activation strategies tend not to be like super efficient, so you won't get 100% on either one. But it could be problematic too. So I think the short answer is it's a great question. It is a question that is going to have to be addressed before we are able to take that into the clinic. And we're going to have to understand how doing any of these genetic therapies, gene activation, is different in deletion versus UPD, and potentially tailor it accordingly.
Another genetic question. That last project you talked about, the researcher is trying to make it so that there's regular maintenance, essentially, so that the treatment continues to work for a long period of time. Most genetic therapies that we hear about in the news they're single time treatments. You take the treatment once and you're done. Is that not going to be the case for Prader-Willi syndrome?
Well, I mean, most genetic therapies are single-time treatment, and many of them also face this issue of maintaining gene expression over the long term. So I could launch into a whole lecture at this point, which I'm sure you don't want to hear. But, most of the gene therapies that you hear about in the popular press are gene replacement. So you take a gene and you stick it in. And depending on the virus you use to do that, there's a particular type of virus, an AAV or adeno-associated virus, that's really good at doing long-term gene expression. It's a bit of a different problem in our case. We're not sticking a gene in there. We're taking the genes that are present and turning them on. So what I think these researchers are anticipating is that may be a problem for us down the line. And this project is kind of trying to get ahead of the curve and understand the issues that we're going to face and address them now. So we do expect, the goal would be to have a one-time gene activation, but we would have to make sure that it is sustained, and that is a different problem than some of the gene replacement activities that you see in other rare genetic diseases.
Second part to the same question, has that ever been successfully done in another disease? This sustaining of the treatment so that it lasts long term?
Well, the whole gene activation field is really relatively new. So for these genes where you're sticking the gene in, those have been successful. And now for several genetic disorders, we've seen that gene expression can go for years. But for where you're kind of turning the genes on that are sitting in the cell already, it's only been possible in the last, like the Gershbach lab really, and a couple of others developed this technology within the last 3 years. So it has not been tried in humans yet, and there's a limited experience even in the animal models. It's kind of brand new technology, and it's very exciting, but they're still at the point of sort of tweaking it and making it work, best for each situation. So we're kind of excited that they're optimizing it in PWS as kind of their first go. So that's exciting.
So you mentioned in the BDNF study that it would be a single dose. Is that just because it's research and they want to observe a single dose? Would additional treatments be necessary if this was a long-term fix?
Right, so that BDNF study is kind of a different approach. BDNF is not missing in PWS, but what it's saying is maybe if we add some extra, it'll address this problem. So it's not addressing the underlying deficit. It's more of like addressing the symptoms. So with the vector the virus that they're using, the AAV vectors, they do long-term gene expression for years. They can direct gene expression for years. So the idea would be that it would be a single administration and that it would last for years. But at this point, it's really proof of principle to say, okay, is this approach of increasing expression of BDNF, because individuals with PWS do have some BDNF, so this is just kind of upping it. Is that a reasonable approach, and does it have a beneficial effect? So we're just kind of at that first point along the development of that as a therapeutic.
Mm-hmm. So you've talked about 3 genetic therapies now. I believe you said one of them required an injection. Where do these injections take place? What part of the body are we targeting?
Ultimately, these would likely be into the spinal fluid to get. So almost all of these therapies, we're presuming that it would have to be genes in the brain that are turned on, and so it would be gene therapy into the brain, which in other genetic diseases now is being sort of optimized. So for Angelman syndrome is actually starting some clinical trials this year using gene therapy of the UBE3A gene, and there are other diseases like Batten disease and other diseases that are SMA that's really life-threatening very early on, where the technology of administering these viruses into humans is being developed. So the idea is that we will be looking at these questions of how can we turn our, the PWS region genes on, and how do we maintain that expression, and how do we do it precisely? And in parallel, in other disorders and in the gene therapy field in general, there are groups that are working on optimizing gene delivery and methods for getting the genes into specific parts of the brain. And in the end, those things will be going in parallel, and the idea is that it would come together several years down the road, potentially to be a therapy for PWS.
Mm-hmm. What benefits of a genetic therapy do you envision for people with PWS? Are we targeting hyperphagia or behavior? What kind of benefits do you think we could expect?
I think we don't know. And I think that's the reason that we're supporting other studies. So there's a study by Dr. Grzechnik, for example, that is looking at, so he's engineered a mouse that just the genes get turned on automatically at different stages of development. And, that project is really to start to understand some of these issues. If we turn those genes on in infants or in adolescent mice, for example, will it have an effect on their metabolism? Will it have an effect on their, you can show in the mice that they have some cognitive defects. Will it have an effect on their cognition? Will it have an effect on their social interactions? So we don't know those things, and we do have examples in other disorders like Angelman syndrome, where they've made similar mouse models and turned the genes on at different times, and what they showed in Angelman syndrome, and I may not be getting this exactly right, is if you turn it on late, you see benefits to some of the motor problems. They have motor problems, they have jerky movements. And if you turn it on early, you see an effect on cognition. And I think we need to do a pretty similar thing in PWS to understand where we're going to get the most benefit, and whether there are time restrictions or whether there are restrictions and like we have to get it into this part of the brain or else we won't see a beneficial effect on this feature of PWS. So there's a lot of work to be done in this area, and it's going to take different researchers addressing it different ways. But it is all ongoing, and I think there's, we'll start to get some answers to these questions soon.
Mm-hmm. So when you state the placenta study may provide a benefit in the long term, how long from now do you mean? And could you please explain what answers you think it will provide? For example, are there druggable targets that can come out of a study like that?
Yeah, I think it is, that one's a little bit more tenuous, like when is going to result in like a clear druggable target. I'm not certain that we're going to see that right away. I think what it's really going to help us understand is the development of PWS, and you might envision in the long term, like understanding if certain genes are not turned on in the placenta the way, if there's deficiencies of like certain growth pathways, that maybe then you would have a therapy. And the question is, would it only be a therapy that's applicable to a developing fetus, or would it actually have application to babies or children with PWS? And I think that is, not knowable at this point in time. I think it's just gaining an understanding of the deficiencies in the placenta potentially, and how that might impact the development of babies with PWS. We know that babies with PWS are born small. They're, about 15% underweight compared to their typical siblings. And so clearly there's some things going on in the prenatal phase already, and whether that is directed by the placenta or not. I mean, you could see a therapy where an individual is diagnosed prenatally with PWS, and then, there's some supplement that they can be given to, help the placenta and increase growth. Whether it would only be prenatal or whether there might be some impact down the line, I think remains to be seen. But it's not something that, it's a longer term project, and it's more of a basic biology and understanding PWS project.
Right. Looks like we have one more question in the queue. Of course, if there's any others, please feel free to submit them. So FPWR has funded or supported Dr. Stormy Chamberlain in the past. She presented at FPWR's Fireside Chat presentation this past fall. Are there any updates on her research?
Yeah, I mean, she does fabulous work, and we continue to work with her. She has recently gotten a large grant from the NIH to continue the work that she started with her FPWR funding, and she's collaborating with Dr. Carmichael, who also was funded by FPWR. I think, they just got a several $1 million grant. So they are continuing that work, which is also focused, it's focused on many things now. It is focused on looking at gene activation strategies, understanding the regulation of the PWS region, which is important for basic biology, but also for therapeutic approaches. I don't think she's published anything very recently since the meeting last October, but she continues to be extremely active, and we are very fortunate to have her have her as a friend of our community, and we will continue to work with her.
One more question has just come in, and that is, how long will the matching gift last for? I can respond to that. The match is happening until the end of the month, so May 31st, and the donor has agreed to match up to $50,000 raised. So again, if any of you are able to donate or would like to fundraise, that matching gift will be in place until the 31st. If anybody has some extra time this month, I would highly encourage you to hop onto the registry and answer a few surveys or update them if you haven't done so recently. The data that you provide through the registry is truly very helpful for the research that's taking place. It's incredibly meaningful. So if you have 5 minutes, 10 minutes, even 15 minutes, if you could just hop over there and answer a survey or two, you'll really be making a difference in research as we move forward. I would like to thank all of you for joining us on today's webinar. It was great to see so many people joining us today. As always, if you do have any questions, Theresa has provided her email here on the slide, theresa.strong@fpwr.org. You're always welcome to send any additional questions her way. She would love to speak with any of you as well. So have a great rest of your day. Please stay healthy as best you all can, and we look forward to seeing you on our next webinar. Have a great day.
Thanks, Susan.